So thank you very much, Paul, for that kind introduction. I must say that I'm very grateful for this opportunity and very excited about giving this talk today. Why do Afrotropical birds breed when they do? Sounds like a question with an obvious answer, that birds breed when there's peak food availability, so that their young can coincide with enough food that helps them to survive. But if you look at that notion more closely, you would see that it's very biased towards temperate ornithology, where seasonality is very correlated with day length and temperature. In the tropics, where there's a lot more dramatic variation in environmental condition and life history, there are so many other correlations that might take place, and so many other factors might be responsible for determining the timing of breeding. In this talk, I will try to share some insights that I got from studying a non-seasonal breeding species, and then what led me to asking the question why birds breed when they do again, and trying to start to answer the question from the scratch. I would like to thank different collaborators who have contributed to this project. Some of them as PhD supervisors, some as field assistants, and even master's students who have worked with me in the last few years. So the question about why do you even invest time studying a common species, like the common bulbul? So for me, the value is that it allows me to shift my focus from trying to find that rare bird to actually focusing on the ecology and trying to understand why the bird does the things that I see it do. And I can find this species in different places and in different environments, and ensure that I have the large sample size that I need to answer ecological questions. This talk will be divided into three main parts. First, we'll try to understand how non-seasonal breeding fits with moult. We're trying to understand how the annual cycle is organized. And then secondly, we'll then test what actually explains the variation in immune function, whether it's the occurrence of annual cycle stages or the changes that occur in the physical environment. And then we'll see how that gets us to asking the question, why do birds breed when they do, and how we try to answer that. So first of all, I want to dispel the notion that tropical environments are not seasonal. The tropical savannas are very seasonal, have a clear wet season and a clear dry season. The notion of non-seasonality mostly applies to tropical equatorial forest, where we assume that there's a year-round rainfall, and so conditions are seen to be stable. But even those are seasonal in their own respects. But I'll show you why tropical savannas are very seasonal, and even quantitatively so. So here we see clearly 2 years of data. We see a clear wet season and a clear dry season, and even variation in temperature following a consistent pattern over the 2-year period. But then why study a species that breeds year-round? For me, it allows me to then sit back and ask the question, how is the annual cycle actually organized? We are tempted to think that breeding should come right after molting, or molting should come right after breeding. But when we see breeding taking place at different parts, then we are really forced to ask the question, are these activities causally linked? So then you ask where should molt be? How is it related to food resources? How is it related to pathogen pressure? How is it related to the occurrence of vectors and parasites that might make birds sick at different parts of the year? So essentially, by having breeding taking place in the wet and the dry season and under different resource conditions, you're then able to ask what environmental conditions actually affect breeding. So I did most of my studies in Jos. So you actually around the AP11 is Ornithological Research Institute, which some of you might know. Haplori sits in central Nigeria. So if you're moving in Nigeria from south to north, somewhere around the center, you find Jos. And in Jos, we have a clear wet season and a clear dry season, each lasting for about 6 months. But then the common bulbul is able to breed both in the wet and the dry season in this environment. So how does breeding fit with molt when it's actually non-seasonal? Okay, so we went out every day and tried to catch a common bulbul, at least one. So for every bird we caught, we checked whether the bird was breeding or not, whether the bird was molting or not, whether this bird was a male or a female, and we took morphometric measurements, body mass, tarsus length, wing length. We aged these birds we caught, and we did that for 2 years, from February of 2014 to February of 2016, and we pulled this data together. And what we found about breeding seasonality is that there could be a breeding bird at any time in the year, even though we see a little bit of a concentration of breeding at the interface between the wet season and the dry season, but pretty much a breeding bird can be seen at any time. But then molting was almost entirely constrained to the wet season, and this was consistent between the 2 years when we caught birds. So one thing we see clearly from here is that despite the weak seasonality of breeding, the seasonality of molt remained consistent, and so that then we can draw the conclusion that breeding and molt seasonality are not causally linked to each other. But what's even more interesting is the fact that these are the same individuals that are capable of breeding both in the wet and the dry season, because our study individuals were color ringed, and so we could tell whether it's the same individual that we see breeding in April and November, for example. So it's not a question of different breeding populations, but the case of individual flexibility in the timing of breeding. But another interesting observation is the fact that the breeders in the dry season carry extra body reserves. So the birds that breed in the dry season are heavier than the birds breeding in the wet season. And we interpreted this as an interrupted foraging response, where birds carry extra body reserves and are able to withstand the impoverishment that's associated with breeding in the dry season. So even if it takes them a longer time to find enough food, they don't starve and can spend the required amount of time incubating their eggs and attending to the nest, and so they're able to maintain both wet and dry season condition, dry season breeding when they have to do so. So the question about immune function is a question of what really explains variation is in immune function. Is it the breeding status or the environmental condition? And clearly this is not a question we can answer from a temperate region where breeding takes place in a particular environmental condition. There are two main hypotheses governing the studies of immune function in the wild. So first is this antigen response hypothesis, where we expect immune function to vary according to pathogen pressure. And then the second is where we expect immune function to vary according to resource availability. And when I talk about resource availability here, it's not just the amount of food available in the environment, but also the fact that the resources that can be used for immune function can be constrained if birds are breeding, molting, or migrating. So the occurrence of another annual cycle stage can limit the amount of resources available to breeding. So birds are expected to trade off between investing in immune function and other annual cycle stages. So that when birds breed, for example, we expect immune function to be low. Okay, so if you consider these two hypotheses, so making prediction based on the antigen response hypothesis, for our own system, we would expect immune function to be higher in the wet season compared to in the dry season, because obviously we expect more mosquitoes and more ticks, more black flies and vectors, other vectors to be transmitting infection. We also expect more environmental productivity, leading to more bacteria, fungi, and all other things that might cause infection to birds. So we expect a clear difference between the wet season and the dry season. But then if you bring the resource constraint hypothesis on board, then you would expect breeding birds in both the wet season and the dry season to show a lower immune response or a lower immune function, because some resources are invested in breeding, and so there's fewer resources to be invested in immune function. But then there are two other possible scenarios where the effect of breeding might only apply in the dry season when resources are actually limited. But the birds in the wet season, whether they're breeding or not, might be able to fund both immune function and and breeding, because there's enough resources in the wet season to be expended. Then a third possible scenario there is that pathogen pressure might be so high in the wet season that it's only birds that are not breeding that can match pathogen pressure with immune function. And so in that case, you see that the dry season breeding and non-breeding birds will be similar in their level of immune function. But then in the wet season, you see a decrease in the breeding birds who don't have as much resources to match pathogen pressure. But we would see whether this is the case for our system when we look at birds in the wild. Interestingly, we found the direct opposite. Immune function was higher in the dry season compared to the wet season. And this was consistent even in individual birds. So it was not just a population level pattern, but for the birds that we caught repeatedly in the wet and the dry season, we see that birds had a high immune function in the dry season and lowered it in the wet season, both for haptoglobin concentration and hemolysis titer. Haptoglobin concentration is a measure of inflammation. So like when birds are fighting infection, they elevate their haptoglobin concentration, while hemolysis titer is kind of a measure of an individual's capacity to destroy infection. And so it kind of tells you how capable an individual is to fight foreign cells. But as I know this point lying here would attract your attention, but then if you think about it, so a bird caught in April, just at the start of the wet season, and then retrapped in November, which is just at the end of the wet season. So it's very possible that conditions local to this individual might still be wet and dry in November and April respectively, and that's what's causing this different pattern. Okay, so if you then consider the difference between breeding and non-breeding birds within the wet season only, for the same haptoglobin concentration, which is a measure of inflammatory response, and hemolysis titer, which reflects the capacity of an individual to fight infection, you get a little bit more clarity, but then also slightly contrary to what we predicted. So for hemolysis titer, we see a consistency with what we predicted, that the non-breeding individuals have a high immune function compared to the breeding individuals. But for the hemolysis titer, we actually see the opposite, that the breeding birds actually have a stronger capacity to deal with infection than the breeding birds, which is opposite to our prediction. So why would breeding birds have a stronger immune function compared to non-breeding birds when they should actually be investing more in breeding and have less to invest in immune function? So we can try to bring these two results together here and try to make sense of it. So if you consider that the dry season is actually impoverished, with birds coming together and several species coming into contact with each other due to limited flow of water, slim resources, fewer fruiting plants, fewer seeds available, and this increased contact during the dry season is likely to increase transmission. And also, the impoverished condition of dry season also may be likely leading to poor body condition, making birds even more susceptible to infection. Then it might make more sense that immune function should be high in the dry season for two reasons. So either birds responding to infection because they get more infected because of higher transmission, or actually birds elevating their immune function so that they are able to deal with the infection that they expect in the dry season. So that then, what leads to a higher infection risk or even immune function is not high environmental productivity leading to more vectors and pathogens and parasites and all other things that might cause infection, but the transmission factors in the environment that's likely to make birds more sick. Then if we look at the breeding birds too, if we think that the condition of the female is actually, even the male, that the condition is important for breeding decision, then we might actually just come to the conclusion that it's only females in good condition that actually breed, and immune function is a measure of individual condition. So individuals who are not capable of fighting infection simply don't breed. So immune function then becomes one of the factors that might explain why birds breed when they do. So that gets us back to the question, why do birds breed when they do? Is it just because of food availability for the young? And that you cannot answer from a single species. So I decided to start all over again. So in 2021, Claire Spottiswoode introduced me to an egg collection. This egg collection was done by Major John Colebrook Robgent. He was a tobacco farmer in Chima, Zambia. So he was a very, very, very special egg collector. He had detailed information of every egg that he collected. And this information includes things like weight, height, mass, volume, dates, location, even information about the adults. And he did that for about 4 decades. So he started collecting eggs in Chima from about 1969 to 2008. And during this period, he collected over 21,000 eggs for about 320 different species. So this is an amazing resource. And all what we did was to digitize this egg collection data and use that to work out the seasonality and and the time breeding for different species, and even with the possibility of checking whether this has changed over time. So let's look at the weather pattern in Choma, which we hope to lay side by side with the information we get from the egg collection. So Choma is in southern Zambia, about 16 degrees south of the equator. It's a flip side of the equator with Jos, and the environmental conditions are pretty similar, only that the timing of the wet season is slightly different from what you see in Jos. Actually, it's opposite. So the rain starts around October, November, and lasts until April, May, whereas in Jos is starting in April and lasting until mid-October. So you can see that it's a simple, clear, single wet season, single dry season, with the coldest part of the year in July. And we intend to check whether the seasonal breeding pattern of the different species from the egg collection actually match this weather pattern. So what we did with the majors data is we calculated the breeding initiation date for each species in these 320 species we had, and we plotted this and look at it. You can see the side by side with the temperature pattern and the rainfall pattern from January to December. You can see that there's a clear bimodal pattern. Most species initiate breeding just before the rains, and just around the period when there's a temperature rise, and then the numbers drop, and then you see another spiking in January. But let's put this side by side with the peak breeding month of each species and try to interpret this together. So with this second parameter, what we did was to calculate the peak breeding month of each species. So for all the over 300 species in the data, we calculated when the peak breeding month of each species was. And then we plotted the number of species with their peak breeding month in each calendar month in the year. And here you see the same clear bimodal pattern, with more species having their peaks in September, October, coinciding with the period just before the onset of the rain, but then also with the period of the highest temperature in the year. And then you have a second peak coinciding with the period just after the peak rainfall and the declining temperature in the year. But then what species are actually breeding here? We see clearly that the species in the big breeding peaks include things like the fork-tailed drongo, the black-collared barbet, the Zambian barbet, bee-eaters, orioles, nightjars, kingfishers. And then a bit much later, going towards November and December, you begin to see the robins and the cystic colors actually kicking in. And then in January, you begin to see the prenia. And then the other smaller breeding peak, which you see, then you see a lot more granivorous species like the widowbirds, the bishops, the finches, and the weavers too, kind of species that build their nest with wet grass and also eat grass seeds. So these species are in that second small breeding peak tend to wait for the peak rain before they hit their breeding peak. And then it feels like if you look at the whole kind of pattern here, you would easily think that the species in the first big breeding peak actually depend on the rain of the previous year or the rain of the previous wet season, while the species in the smaller breeding peak depend on the rain of the current wet season. And then at face value, if you look at the data, you also think that there's a nice correlation between the diet or the foraging behavior of this species, how it relates to rainfall and the breeding pattern of the species. But then if you look at it more closely, you see that there might also be some difference in the sizes of birds that breed at the different periods. And then the nest architecture of this species might also be important, because the species in the large breeding peaks, like the drongos, are like open cup nesting species. Then the barbets and the barbets, the kingfishers, and the bee eaters are like cavity nesting species, which either nest in tree cavities or even cavities on the ground. And so these types of nests are likely to be sensitive to a lot of rain. And then the species in the smaller breeding peaks are mostly like bottle-shaped nest turtles, which have much more covered rain, which are weaved with grass, wet grass and all that, and that might not be affected by rainfall. So it's not obviously clear why a species might be breeding at a particular time, and whether this is just a balance between the different factors that drive the timing of breeding. But then if let's kind of cut the year in a bit more intuitive place, because January to December doesn't really mean anything to a bird. But then if we look at it in terms of the environmental condition, July is the coldest part of the year and in the dry season, and we see clearly that's the part of the year where breeding peaks are at their lowest. So if we assume that the year starts at that time, or even ends at that time, and so we cut the year then and stitched it, then we see clearly that the peak temperature in the year kind of precedes the rainfall. And then if you put the breeding data together, we see, and each dot here represents a species, and then on the y-axis you have the duration of breeding, or the duration of the breeding period for each species, and then the peak breeding period for each species. And here you see that there's a cluster of seasonal breeding species here and here, and then several other species seem to be doing something interesting. What you see clearly is that the parameter space is fully occupied. So what we intend to do in the next few years, in the next few months, is to determine the environmental, so to kind of the ecological and life history factors that predict when a species is breeding. So a species position on the parameter space. And currently what we're doing, we are analyzing insect availability and analyzing grass seed availability. We're checking nest architecture. We're looking at body size. We're looking at phylogeny. We're pulling all this information together, and we include rainfall, seasonality, and all. And then we try to see which factors explain a species position on a parameter space. And then we'll also put information side by side, so the breeding curve and the resource curve, and test whether the food availability curve precedes the onset of breeding, or whether it precedes the occurrence of juveniles. And that allows us to know whether the timing of breeding selects for for the condition of the adult or the survival of the juvenile, or even kind of tries to balance between the two. And then finally, what we will be doing is also to do a manipulative experiment where we increase breeding workload in the wet season and breeding workload in the dry season, and try to see how that affects immune function and an infection risk, survival of juveniles, and kind of the breeding outcome overall. And that tells us whether these factors play an additional role in the success of breeding depending on environmental conditions. I would like to thank all the organization and bodies that have supported this project over the last few years. And at the same time, thank you all for listening, and happy to take your questions. Thank you.